pleasure to be here. As people know, NIH is very invested in trying to develop better treatments for people with traumatic brain injury, preventing traumatic brain injury, helping people recover from traumatic brain injury. And we have been in this business for a long time, and so we bear the responsibility for the lack of progress that we've seen, as well as the progress that we've seen. Clearly, I think we know, the people in this room know what the challenges are, and I'll talk a little bit about those. But I think also, in terms of the Massey Foundation, what it can do, I think a lot of people in the room know that NIH has funds to do high-quality traumatic brain injury research, but to get those funds is an incredibly competitive process. And that's because we have many, many different diseases, and they're all suffering from the same lack of funds. But the good news is we have a lot of really good people coming into the field trying to work out these problems. And our system in the United States, as compared to other countries, is very much a democratic, peer review system that is highly competitive. And I know from my work outside, prior to coming to NIH, that to get an NIH grant might take 5 years of work. And where does those funds come from? Well, oftentimes, if you look at the history of people's successes, the funds come from groups like the Massey Foundation, who get people off, who get the preliminary data that's needed, who get the organization started. It allows you then to compete for the NIH funds. So when I talk to nonprofit organizations, except for the really big ones, I generally tell them what your best bet is probably to invest your funds in your group and your disease that allows you then to leverage for NIH funds coming down the road. And so that's why I think this is really important, to have a group that gels, that has some funding to get things off the ground, is really what we need at NIH. We can't, unfortunately, fund all those seeding efforts just because we don't have enough funds to do that. So we've all been in this business for a long time, but actually protecting the brain from traumatic brain injury goes back probably to the beginning of time. So Adam and Cain times, that's the first brain injury I can remember. So at NIH, as people know, as I mentioned, we try to kind of spread our funds across a wide spectrum. So we try hard not to bucket in any given one area. And that spectrum can be described as basic, translational, and clinical research. And we have to pay attention that we are doing the best for each of these three major categories. And as you know, the basic research is giving us the foundational knowledge that can move to translational research, where we develop things that look like they're going to be helpful for diseases, and then we go test them in clinical research. But as you all know, it's not a one-way street. There's a lot of crosstalk back and forth, and in certain areas, because of the burden of disease, we take these leaps of faith where we think, okay, we know something about the basic research. I realize I don't know everything, but maybe this is going to show something in the patient, so we move it into that translational zone, and then into the clinical zone. And I think that, over my career, what I look back at is that sometimes you get really lucky. You find something that just cannot lose. So I think one example is the mutation that was found in people that affects their lipid metabolism, so their cholesterols are 0, and it's due to a missense mutation, and they don't get any coronary disease. So here you have people, a natural experiment, the mutation is gene, and nobody gets coronary disease, and you know it's related to cholesterol. So all you have to do is develop a drug that knocks out that gene product, and you know that's not going to be harmful because there's people living completely normally without the gene, and it's going to drop your cholesterol. That's the story of the new drug out for cholesterol lowering. Or the other example being the orexins and sleep. So why do we sleep? How do we sleep? People figured out this orexin system is really driving sleep. So it's pretty clear a drug that's going to attack the system has got to work. I mean, you're basically getting into some pathway that's really kind of narrow, and you can make that jump to the clinic and be pretty assured that you're going to be successful. traumatic brain injury, so far we're not in that narrow pathway. We're dealing with an incredibly complex problem, and we've tried to kind of take this giant leap with a single drug across this big divide, which is from the basic to the clinical, and in the acute stage situation we really haven't been successful. And so I'm going to argue that we need to kind of break down that process instead of taking the big jump, the magic pill, to try to break down the physiological processes and try and attack one versus the other, one and then the other, and go stepwise to a process rather than taking these big leaps. And the key to being able to do that is to actually be able to measure in humans what you think the therapy is supposed to be doing. And I think that's been, if I had to look back at what we've done, I think that's been our biggest mistake, that we haven't really we haven't been able to actually identify exactly what the drug should be doing if it's working in the humans. Lots of reasons for that. But if you don't have that ability, then you're treating the brain as a black box. You're giving the drug, you're hoping that it did in the human what it did in the animal, and then you wait 3 to 6 months, see what happens. And that hasn't worked. And I think NINDS, not just for traumatic, this hasn't worked for anything. It's not just traumatic brain injury. It hasn't worked for Parkinson's disease, Alzheimer's disease, any of the diseases that we go after. We're looking at protecting the brain. That strategy does not work. So what we would like to do is to see people work in this field to develop actual measures that you can look at humans that are reflective of the pathophysiology that we think is important, and then develop therapies and test them, did they actually do something in that space or not, rather than going on to big phase 3 clinical trials without proof of what we call target engagement. So that's kind of I think that as a director, that's probably the change that I'm going to be looking for as we move forward. And again, as I said, it's not just traumatic brain injury, but it's all of our clinical trials. In terms of TBI research, this is kind of what our portfolio looks like. So most of the money is going into treatment in the acute phase of the disease. Still, that's the thing that drives people into this field to begin with, and certainly, I think as we heard last night, is a driving factor for the Massey Foundation's interest in traumatic brain injury. But you can see that the recovery piece gets quite a smaller amount of funds, and it's a much less crowded field than the acute injury field. We do a lot of work in the mechanisms of acute injury, some work in diagnostics and biomarkers. So that's kind of what the portfolio looks like. Most of the funding is coming out of NINDS. It's about $62 million a year. There are some other funds that are coming from other institutes. So NICHD is Child Health and Development. So they fund traumatic brain injury research now. Mostly that money is going into recovery from traumatic brain injury research because the NICHD houses the National Center for Medical Rehab Research. And so they do most of the rehab research, and not simply on children. Actually, most of it's on adults. So that's where the other part of the money comes from. And so if you look at the burden of disease due to traumatic brain injury and you look at this number, it's really, they don't add up. This is a very small amount of investment for traumatic brain injury. And so people come to me and they say, well, why can't you put more money in traumatic brain injury? But then say, well, everybody comes to me, I always show them this figure and the number never matches. So if there's no disease I know of that we care about, that the amount of money we invest comes anywhere close to the burden of disease. So that argument is a completely failed argument because it doesn't really help us. You're just stating the fact that that you're not getting enough money, and I say fine, but no one's getting enough money. And that's really the problem that we've had. And as you know, the NIH budget has been flat for 10 years, and so we can't actually do anything. There's nothing more. There's no more money that we can put into anything because all the money has basically been divvied up into the different diseases, none of which are getting anything close to what they need. Our research, as I said, kind of covers the the spectrum of different diseases, different types of traumatic brain injury research. We are doing something different that Jeff Manley knows quite a lot about, and that's we decided to kind of break our policy to fund large natural history study in traumatic brain injury that would look at the heterogeneity of traumatic brain injury with regard to how it presents as well as how it's treated, with the idea that there was enough sophistication known about outcomes that we might be able to match up different types of head injury with different therapies, and at some point try and do what we call comparative effectiveness research based on prospective data collection. And so we have two projects, Jeff's project, Track TBI, and Michael Bell's project in kids called ADAPT. And those are prospective data collection studies trying to look at comparative effectiveness. And the reason I say that that has not been our policy is because we have about 200 to 400 diseases, and every single disease wants to do that. We just cannot invest all the money into these kind of prospective data collection studies. Because the risk of not, at this point in time, the risk of not getting data that's going to inform treatment decisions, move the science forward, is way too high. Like the investment currently is thought best to move into actually direct hypothesis testing. But this is a pilot, and hopefully we get proven wrong, and that this area of doing research changes over time. There are certainly statistical methods in which you can look at prospectively collected data and make fairly strong conclusions, statistically based conclusions about causative relationships in those data. But it's got to be a very statistically rigorous process to do that. And so we actually recently had a discussion with the neurosurgeons in the United States about moving in that direction to look at the efficacy of neurosurgical procedures because there is very little scientific evidence backing many of the procedures done in the States and around the world, and doing clinical trials to actually figure this out is probably a hopeless quest, but there is potentially ways of going about it in this other fashion. So Track TBI and Adapter, I think, important for all of NIH in terms of how we think about doing studies forward, but it's particularly for the TBI field. So we have about 160 extramural grants in FY14. If you look now compared to 10 years ago, there's much more studies in the concussion space than there were in the past, where most of the studies were on the acute, moderate to severe types of head injuries. There's now significant more interest in looking at concussion. That's driven in part by a couple of things we'll talk about. Certainly one of them is the issue that is facing the United States right now, which is, do parents let their kids play contact sports? So I'm sure people in the room have kids, they talk to other parents, and this is a big concern across the United States. And it's fueled by, on the most extreme case, by reports of chronic traumatic encephalopathy, which is a degenerative, really bad degenerative brain disease that occurs after multiple concussions. But again, what's the evidence? Should parents be worried that their kid playing football is going to have CTE? There's really no evidence for that at all. But in the public press, it's all one thing, and so then the parents come, well, what is the evidence? And then, well, we don't have any evidence, basically. So I think that's also that's driving concussion research. In addition, the DoD, as you all know, has usurped NINDS as the major funder of TBI research over the last 10 years. So we think they've put in about $1 billion over the last 10 years, where we're 82 a year. So we're about 820 million. They're probably at $1 billion. And we know that they are driven by the problems that they've had in Iraq and Afghanistan with blast injuries. Again, something we know very little about. But also that got them to look at TBI in general, which is rampant throughout the military, given the age group of the people in the military. And 90% of those have nothing to do with blast. They are the type of concussion injuries that we would see in the civilian population as well. But anyway, the DoD really kind of took that, really proud of them. They took that up and they put a lot of money into research for traumatic brain injury at that mild level, the blast level, much less at the severe level, but some. So track TBI. The other thing that track TBI allowed us to do is to actually work with an international group. So the story of this is kind of interesting. Before I came, NINDS decided that in traumatic brain injury, that there needed to be common data elements so that when people did research, they would collect data in a similar fashion, and they picked traumatic brain injury as the first thing to go after. And Andrew Maas in the Netherlands was actually given a grant to start working on this, and probably, it must be 10, 12 years ago when it got started. And of course, it went very slow in the beginning, but then the steam started rolling on it, and we got groups together, and we locked them in the room. Some of the people were here, getting locked in a room. We wouldn't let them out until they all agreed on what common data elements they would use. And they did it. They came out with common data elements for traumatic brain injury research. They got published. And it was an international U.S. effort. So we had this opportunity then to actually test this idea I mentioned before, which is the prospective data collection, in a fashion that would bring the European groups in, bring in 10,000 patients, bring in the U.S. groups in with thousands of patients. We really had the opportunity to do something in this prospective data collection business that we could never have done before. A lot of it because of this potential for the international collaboration and bring the numbers up in terms of that project. So really excited about to see how that's going to turn out. So I think we probably get something around 2018 or 2019. And then the Canadians have been involved. There's a group that One Mind for Brain Researchers work with called the Interbeer. So we're growing this international group to do TBI research in a cooperative manner. The DoD, as I mentioned, very interested. They worked with us with the common data elements to develop a data repository. So common data elements are great because people do research using similar languages, but then the question is, it's not really that helpful unless the data goes somewhere that people can see it. And so we worked with the DoD to set up what's called the FITBR, which is a federal it's a repository for traumatic brain injury research. And both the DoD and NIH have come together, and we will not fund any TBI research where the data does not go into FITBR. And it's got to go in every 6 months, and it can be embargoed until the time frame that it can be opened up. But this is, again, the investments that were put in were unusual investments, and the data on these large number of patients, the taxpayer putting money into this, it's only going to be valuable if this data is available not just to the group that's collecting the data, but to others who can look at this data and try and understand it or add to it in the future. So a living type of database. It's very expensive. It's costing about $2 million a year to run this. It's a real tough thing to get your data in. But it is kind of I think this is the future of where NIH is going to be going with all of its projects down the line. The problems now are mostly logistical, but the concept that when the taxpayer pays for data collection, the data collection is going to be publicly available, that train has left the station. That's not coming back to the station. And so we're kind of out in front trying to put this together. It's clearly got its imperfections, but I think it's been a very good start. So crossing the valley of death, I talked a little bit about this in the beginning, the idea of how do we get past our histories of trying things, they don't work, and we're not really sure why they didn't work. So I think you want to, as you go across this valley of death, you want to pick a good horse. So you want really high quality preclinical data. I don't know if people are aware, but there's a lot of angst out there because when people have looked at preclinical data, they found oftentimes it's not reproducible. And this we knew about this for a while. I can tell you exactly how we learned. But the point now is that everybody knows about this. It was published, Nature and Science, and if anything's going to threaten the NIH budget, this is what's going to threaten the NIH budget. If Congress believes that NIH is funding research that's not reproducible, they could cut the budget in half tomorrow. This is probably the major, biggest concern at NIH at this point in time. And so why is it not reproducible? It's very complicated, not simple. It's not due to people faking things. It's due to lots of different factors. I can tell you the example that we had in spinal cord injury. We ran three or four centers that would gear up to reproduce results that other people's labs had shown that a certain drug or agent or process was protective in spinal cord injury. I think they tested 11 compounds. None of them reproduced. Zero. So what was going on? Now, in the drug company, they just say, I couldn't reproduce it. They publish this in Science. I can't reproduce, 70% of what NIH, funds. I can't reproduce. So what this group did, though, is they were, they wanted to know what the problem was. So they went back and they worked with the investigators who were doing the spinal cord injury work. And they said, well, I couldn't reproduce what's going on. And they got together. They kind of shared how they did their experiments. And then they were able to reproduce the results. They were never able to get the effect size that was originally published. But in most of the cases, not all the cases, they were able to see some signs of protection, but it was never as big as what was initially published. So I think effect size is probably something that we haven't really paid attention to. We pay a lot of attention to p-values, statistical significance, but the effect size and the generalizability of a result, I think, is really, really important to pursue. So when we have a therapy that we think is going to work in traumatic brain injury, we should spend at least a year to figure out, well, when does it not work? Not just keep doing your experiments and say it works, it works, it works, it works, it works. Try and test, when does it not work? How long after the injury can I actually get away with it? How old do the animals been? What the sex of the animal have to be? Try and pursue not just the conditions in which it works, but the conditions in which it doesn't work. Because that robustness, when you go from one strain of animal to another, you lose effect, you go into patients, then the heterogeneity is gigantic. So I think we have to think about generalizability and robustness or effect sizes as much as we have to think about statistical significance. And then the good laboratory practices of, blinding the assignment of animals to the therapy or the control group. That should be a blinded assignment, blinded outcomes. Trying to do everything you can to set up the experiment so that these little inklings of bias can't kind of change things. And I think the business about getting across this divide, if we try and jump across this river, we don't know how far we have to jump. And we've tried to jump all the way to the other side. And sometimes I think you got to build a bridge, or you got to put some stepping stones in the water so you can get across one way or the other. And I think the way to do that, as I mentioned, is to try and develop things that you can measure in humans that then your drug or your therapy, when it's given to humans, you expect to see some change in that marker should the drug be doing what you think it's doing. And to mark things out in stages. So spending a lot more time in phase 2 work before jumping into phase 3. So we have programs. So at NINDS, we didn't just say you guys should do this. We actually have programs to do these kind of things. Programs that we have an Office of Translational Research, which is to bring therapies through this process to go to the FDA for an IND and then into phase 1 studies. So we will put money behind these programs to get therapies out. But these are not your usual R01 grants. These are managed projects, and they're managed by people who came out of pharmaceutical industries, and so they have milestones just like you would have in a company. You do this, you do that, and if it doesn't work, it's done. The money goes away, and you have to retool. And so one thing that was bad for NINDS is that a lot of the pharmaceutical companies got out of neuroscience development. So we had to really put this program together. You know, companies can say, I'm not doing stroke anymore, but NINDS can't say we're not going to do stroke therapies until the companies come back. We have to make that effort as best we can. So we have programs. The good news for us is when the pharmaceutical companies got out, they fired lots of people, and we were able to hire really good people from pharmaceutical companies into NINDS to run the programs. But these are milestone-driven, really kind of very much like pharmaceutical advanced programs. And then we have a phase 2 network set up to do first-in-human studies called NeuroNext. And this is not disease-specific. It's not neurology or neurosurgery. It could be emergency medicine. It could be any professional group. Anybody can apply to get into this network. They do just phase 2 studies, biomarker-informed phase 2 studies. They are currently running 5 simultaneous studies, and it looks like it's going quite efficiently. So this is available if people wanted to jump in with a therapy to test. They'd have to gear up. If you're going to do severe head injury, they'd have to gear up, get their neurointensive care people involved. But that's what they've had to do so far. They've done diseases in kids and infants. They've done diseases in adults. So that's what they do. They kind of move around, get the expertise they need depending on the trial. Okay, we support a lot of resources that people should be aware of. The Toolbox, which is a standardized neurological assessment tool. PROMIS, patient-reported outcome tool. These are all free. The PROMIS tool, I believe, you can set up your own patient-reported outcome system through the NIH program, and you can run it, getting your patients on the web to put in patient-reported outcomes. And these patient-reported outcomes are unbelievable, highly replicable. This was not done by neuroscientists. This was done by people who were experts in educational testing. So the guys who put together the MCATs and all those other tests, they know how to do testing, and they put together these tests, and they're incredibly statistically valid in the ability to compare people with regard to these different scales. The Connectome project for brain injury, very important. So we fund a lot of imaging research. People come in, they have 25 controls, 25 affected. They think they see a difference. How do you know the difference is really just a variation in normal? You have no idea. The Connectome project brings in data from thousands of people on the connections in the white matter between different areas. And so if you did a study and you used the Connectome acquisition parameters, you would be able to compare your data to 1,000 normal people. And then you would know if you really have something or not. I think that would also help, particularly in the imaging field, to get more replicability. People may know the imaging field has really been held back by the inability to replicate a lot of the work that people do. They can get it in their one lab, but no one else can get it. So diffusion perfusion imaging, which I worked on for years and years, it's not an approved diagnostic because it's never been validated. We're going to be doing a stroke study, hopefully to validate it for the first time, but it's been 20 years. Okay. I'm going to end up with the sports concussion issue. Again, as I mentioned, it's a big problem, and this is kind of my little favorite thing that I've seen with regard to concussion. You know, people say concussion is a functional problem. There's no actual anatomic damage in the brain, but I think that was true because we didn't have the ways of looking into the brain to see if there was damage. But this is an interesting lab that studies viruses in the brain and how they affect the blood-brain barrier and how inflammation traffics across the blood-brain barrier. What they do is they kind of smooth out the surface of the skull so you get a thin skull, and then they can bring a microscope down and see right into the brain with a confocal microscope. And they had a student, a summer student, post-bac student, maybe it was yours, I don't know, who was a little clumsy with the microscope, and he would just basically touch the surface of the skull with the microscope, but then he looked and saw what happened, and it's pretty amazing what happens. So this is Dorian McGavin's lab. So when you touch the surface of the brain, so that's the control. This is what's happening to a dye in the blood vessels. It's basically it's leaking out of the blood vessels. And so this is, I think, what concussion may be. This is very surface of the brain, a slight touch to the surface of the brain, and you see these changes in the blood vessels. And then you see this glial response. On the left side, you can see the glia changing their shapes into what they call jellyfish glia. And these are microglia that then are moving throughout the brain. And what they're doing is actually plugging in holes. So when you have this little bit of injury, you can follow the glial cells on the surface of the brain. You see some of them die. And then you can see these other glia come in and plug the holes. So I guess the idea here is that what we've been actually looking at in the brain in terms of traumatic brain injury has often been static pictures at one point in time. And now we actually have amazing new technologies where we can look at the brain over time and actually see the movie of what's going on. I think it really offers us an opportunity to look in a different way and to see things that we could never see before. So tools, I think, are really, really important in trying to move brain injury forward. We really definitely need better tools. The Brain Initiative, which people may have heard about, is the one thing that NINDS has gotten extra money for. NIH has got about $50 million additional money for the Brain Initiative. And the Brain Initiative is focused on developing the tools that will allow us to interrogate and modulate neural circuits in the brain. So, what the patient's symptoms are due to some problem in their circuitry that's not working. We don't have a way of actually recording that circuitry or the problem in the circuitry. We're stuck with structural type of information. But what if we could, measure activity in the motor system or measure activity in the attention system and look and see, what happens to people's circuitry after head injury, specifically important circuits, and also to look as they recover. The amazing thing about brain injury is people can recover a huge amount of abilities over a period of a year or so. To understand how that's happening, maybe intervene to kind of really speed that up so that everyone gets a good recovery as opposed to a minority. So lots of things are going on in the traumatic brain injury field. It's an incredibly exciting field. I think I think one thing about the traumatic brain injury field now, it's not naive anymore. It's really a sophisticated field. So it has the opportunity to do things that other fields are going to be, they're going to be getting into this mire of making jumps and leaps in faith. The brain injury field, I think, now can really get down to brass tacks and trying to work out the details of what's occurring in the different types of brain injury and then come in and target therapies when they're needed. So I'm really looking forward to working with the group here over my years at NIH, and I think there's going to be good things that happen. But I think it's all tough. We know it's tough. We need good young people. We need groups like the Massey Foundation to see things. So thanks very much.